Next up, we have Annie Bryant, who is a PhD candidate with Ben Fulcher and Mac Shine at the University of Sydney. After growing up in New Jersey and working in an Alzheimer's disease lab in Boston for several years, Annie moved to Sydney to pursue her doctoral studies applying interdisciplinary methods from complex systems to multimodal neuroimaging data, with the goal of clarifying multi-scale properties of the brain's dynamical structure, with a particular focus on neurodegenerative and neuropsychiatric disorders. Her talk is titled Mapping Direct Information Flow Between Homotopic Regions of the Human Brain. Please join me in welcoming Annie.
All right, good morning, everyone. My name is Annie Bryant, and as of last week, I've submitted my PhD thesis after studying with Ben Fulcher and Mac Shine at the University of Sydney. And I'm here to talk about mapping homotopic connectivity with and without directed information analysis. So for those who are unfamiliar with homotopic connectivity, like myself at the start of this year, I will quickly orient you to the different types of connections in the brain. First up is intrahemispheric, where you have a structural or functional connection between areas of the same hemisphere. Interhemispheric means a connection crossing from one hemisphere over to the other. And then we have homotopic, which is a special type of interhemispheric connection that specifically refers to mirrored homologous regions in the left and right hemispheres. And homotopic connectivity interfaces with a heap of really interesting lines of research around laterality and functional specialization, but today I'll be focusing mostly on the resting state setting. So why should we care about homotopic connectivity specifically? For starters, it emerges early in embryonic development, and it's maintained throughout the lifespan with healthy aging-related changes. It's observed across numerous species, and in addition to humans, including macaques, rodents, and zebra finches, the latter of which is particularly interesting since they ontogenically don't have a corpus callosum physically linking the two hemispheres. And we also know from emerging work that homotopic connectivity is highest in primary and unimodal sensory motor regions, and homotopic region pairs show greater laminar, transcriptomic, and receptor density similarity. And so given our growing collective appreciation for this functional phenomenon, in this project I wanted to dig deeper to understand how homotopic coupling relates to multi-scale properties of cortical organization. And there's 4 key aims that I'll briefly review today and are detailed in our recent preprint. That was the last chapter of my thesis. So first, we wanted to understand how the spatial variation in homotopic coupling relates to macroscopic properties of cortical organization. We next asked whether this phenomenon is robust to disease. We also investigated how homotopic coupling relates to broader cortex-wide functional connectivity. And lastly, we wanted to start exploring potential physiological mechanisms that might be underpinning homotopic connectivity. And originally, I wanted to investigate homotopic coupling through the lens of directed information flow. So we borrowed a measure from information theory called directed information, which is quite similar to Granger causality for those that are familiar, in that it captures how much knowing about the past and present of one brain region's activity helps reduce the uncertainty about the present of another brain region. In computing this measure and averaging the 2 directions per homotopic region pair, we found this really interesting gradient from the lowest values in these limbic association areas to the highest values in unimodal and primary sensory motor regions. Nice. But we also found that this rather complex, nonlinear measure of lagged directed interactions yielded extremely similar values to that of the Pearson correlation coefficient between homotopic pairs, which captures linear contemporaneous coupling and just assumes a joint bivariate Gaussian distribution. And so I think there's some really interesting questions there surrounding the similarity of intrinsic neural timescales between homotopic region pairs and how this timescale might diverge across the cortical sheet, which could justify using more complex measures like directed information. But for now, I've pivoted for this project and indeed for this presentation to focus on the Pearson correlation coefficient for the work presented today. And so I'm now working with structural and functional connectome data provided by the Human Connectome Project and pre-processed by the Enigma Consortium, which we heard about at length in Paul Thompson's keynote talk yesterday evening. And they've provided group averaged structural and functional connectomes that I've worked with for this project. So let's dig into our first main question, which was asking how homotopic connectivity spatial variation interfaces with multimodal properties of cortical organization. As we'd seen before with directed information, we see a gradient in homotopic coupling magnitudes from lowest in limbic and association regions to highest in primary sensory and motor regions. And this pattern of spatial variation is significantly associated with key macroscopic gradients of structural and functional organization, including the anatomical hierarchy based on feed-forward and feedback laminar differentiation, and the first principal component of the transcriptome across the cortical sheet, which is derived from the Allen Human Brain Atlas to capture maximal variance in gene expression and provides a proxy for cell type distributions across the cortex. And note the positive correlations between homotopic coupling and both of these maps, which capture distinct physiological underpinnings but are closely related to and spatially aligned with each other. And lastly, we see a negative correlation between homotopic coupling and the first principal gradient of functional connectivity, which captures a dominant functional axis spanning from unimodal sensory areas to higher order limbic and association areas, often referred to as the putative functional hierarchy of the cortex. And these three trends of spatial alignment here converge on this idea that Stark and colleagues proposed in 2008, that unimodal and sensory motor regions sit in the default state of higher interhemispheric synchrony, while we see decreased homotopic coupling in favor of lateralized hemisphere-specific processing as information is passed up this putative functional hierarchy of the cerebral cortex. Having mapped the spatial variation of homotopic coupling and key multimodal correlates, we next asked whether this phenomenon is robust even in the context of disorders known to disrupt functional connectivity and indeed homotopic coupling. So as an exemplary case study, we focused on neuropsychiatric disorders, which generally show disease-associated reductions in homotopic coupling. So we queried the UCLA CNP dataset, which includes schizophrenia, bipolar I disorder, and attention deficit hyperactivity disorder. And these cortical maps visually demonstrate markedly similar trends in high versus low homotopic coupling relative to controls across the cortical sheet. Though when comparing the difference in coupling values per disorder, we found these heterogeneous disease-associated perturbations that were predominantly negative, meaning reduced homotopic coupling in the corresponding disorder. But these changes were relatively small, and ultimately, the resting state homotopic coupling gradient was still nearly perfectly preserved in all three neuropsychiatric disorders. And this robustness is very surprising, given that functional connectivity in general is known to vary considerably across individuals and from cohort to cohort, even in neurotypical populations, much less in the context of heterogeneous neuropsychiatric disorders. This suggests that this homotopic coupling gradient is a very robust macroscale axis of functional architecture. Since we've established that homotopic coupling is associated with key fundamental axes of cortical organization and robusted disease, we next asked how it relates to cortex-wide functional connectivity. And here we wanted to understand how a region's homotopic coupling magnitude relates to its overall functional connectivity, both within and across hemispheres. And so I've attempted to depict our method schematically, where we collapse across the left and right sides to yield one intrahemispheric and one interhemispheric coupling value per region pair, which gives us this FC matrix shown on the left in panel B. So here the left diagonal contains intrahemispheric connections, and the right contains interhemispheric, with regions ordered according to the mean homotopic coupling values, as indicated along this center diagonal line. And this visually reveals that regions with higher homotopic coupling tend to show greater overall synchrony, both within and across hemispheres. And this indeed bears out statistically when summarized as the average nodal strength, as shown in the bottom right here. So collectively, this suggests that homotopic coupling co-varies with global resting state synchrony across the cortical sheet, where we see more dominant brain-wide synchrony in unimodal sensory and motor regions that weakens as you ascend the putative functional hierarchy towards higher order association regions. And lastly, we wanted to start characterizing possible physiological mechanisms that might be supporting homotopic coupling across evolution and the lifespan. So while members in this room might argue about the extent to which this is the case, anatomical connections can provide a physical substrate for functional connectivity, and evidence from the literature suggests that the majority of fibers in the corpus callosum innervate homotopic region-region pairs. So we compared structural connectivity derived from diffusion MRI with the homotopic functional coupling, and we found no statistical relationship between the two. But importantly, this just means that in this data set, the quantity of fiber counts itself does not relate to homotopic coupling, though it could very well be that properties of white matter architecture are still mechanistically related to homotopic coupling, particularly given that fibers in primary and unimodal sensory motor regions are more heavily myelated and exhibit faster conduction. We also found no association with physical distance between regions, which is in line with previous evidence that homotopic coupling doesn't follow the canonical exponential distance rule to which structural and functional coupling typically adhere in the cerebral cortex. And given hypotheses about vascular contributions to homotopic coupling from Patrick Drew and David Kleinfeld's lab, we also examined the spatial relationship between arterial and venous density versus homotopic coupling, finding only a weak but still significant negative association with arterial density. It's tricky to disentangle biological versus methodological contributions to this association, given that BOLD itself is sensitive to neurovascular coupling, but this suggests interesting avenues for future research to explore the role of vascular geometry in shaping interhemispheric synchrony. So I'll end my presentation with just a couple of open questions that I'm really excited to explore further after this final project of my thesis. First, I think there's a great opportunity to better understand how homotopic coupling, to understand it through the lens of increasing task complexity, which could help us understand if, and indeed how, this coupling reorganizes across the cortical sheet and balances with hemisphere-specific lateralization with greater cognitive burden. And I also very much want to explore other candidate physiological mechanisms, particularly shared subcortical drive, since homotopic coupling could plausibly arise from either direct monosynaptic communication and or entirely independently via shared thalamic afferent connections, or some combination of the two. With that, I'll end there and thank my research groups for support all throughout the thesis, and the organizers for inviting me in the audience for your time. Thanks.